Hi everyone, let's go ahead and talk about microbial genetics, and this is going to be Unit 3. There's a few pioneers in genetics that I want to mention here. These are the pioneers of genetics. And remember, the microbiology pioneers were Anton van Leeuwenhoek, Louis Pasteur, and Robert Koch. And now we have the historical pioneers in genetics, which would be Frederick Mesher, and Phoebus A.T. Levine, Rosalind Franklin, you've probably heard of her, and James Watson and Francis Crick. So Frederick Mesher discovered DNA in 1869. He isolated molecules of substances, including a substance he called nuclein, which was actually DNA. And he isolated this from white pus cells, lymphocytes, and neutrophils. And he was a Swiss physician turned medical researcher. In the 1920s, Phoebus A.T. Levine discovered that it contained phosphates, or 5-carbon sugars and nitrogen bases. Phoebus A.T. Levine was a Russian physician and researcher. He studied mostly the organic structure of sugars. Rosalind Franklin was an English grad student who used X-ray crystallography to demonstrate the double helix. She later discovered the helical nature of DNA. She might have got the Nobel Peace Prize in medicine, but she died in 1958, and they don't award that prize posthumously. In 1953, James Watson and Francis Crick elaborated on the three-dimensional helical structure of DNA. They worked with Rosalind Franklin and were later awarded the Nobel Peace Prize for medicine. DNA is contained within the bacterial chromosome. It's usually a single circular molecule and that is haploid, or in other words, it has one single copy of DNA. It's localized in the nuclear space within the cytosol of the organism. The cytosol is chemically rich fluid inside of the cell. DNA exhibits supercoiling, or tightly wound DNA that fits inside of the organism. Because of supercoiling, there is enough room to pack tons of genetic material into a small space. And then there is a term down there at the bottom called haploid, and it basically means having a single set of genetic information. It's not a pair. Like in humans, we have 23 pairs of chromosomes, but in a haploid situation, there's only one set. Let's go ahead and compare the structures of DNA and RNA. So DNA is a long double helix chain of nucleotides. Okay, a nucleotide is either ribose in RNA or a deoxyribose in DNA, and they're basically a sugar backbone. And they are attached with phosphates to pyrimidines or purines. Okay, the purines that are found in DNA are either adenine or guanine, and the pyrimidines are either cytosine and thymine. A purine is a 2-carbon pentose structure with nitrogen, and a pyrimidine is a single ring of a 6-carbon structure with nitrogen. And the backbones are twisted together in DNA. DNA is involved in the production of RNA. In RNA, thymine is replaced instead with uracil, which is a pyrimidine. RNA is short and single-stranded, and it contains the sugar ribose and not deoxyribose. So here's what the structure of a pyrimidine and a purine looks like. A pyrimidine is a 6-sided structure where each line represents a carbon. Cytosine and thymine and also uracil in RNA are pyrimidines. Purines are 2 attached 5-sided carbon pentose structures, and that would include adenine and guanine. Each strand of DNA is linked by hydrogen bonds connecting purines to pyrimidines. And then the strands are twisted together. So for example, we have a deoxyribose in DNA, and then we have a purine like guanine linked together to a pyrimidine like cytosine, and those are linked together by hydrogen bonds. In this image, you can see the double helix of hydrogen-connected base pairs. So why exactly is it important to have an understanding of bacterial genetics? Okay. Understanding is necessary when learning about the development and transfer of antimicrobial resistance between microbes. We have to understand transfer of resistance. For example, we have to understand the transferring of resistance in organisms like methicillin-resistant Staphylococcus aureus, or MRSA, or even in VRE, which is vancomycin-resistant Enterococcus. We also have to have an understanding because we have to understand how to investigate mutations that alter microbial phenotypes. Mutations like the pigments of Serratia marcescens. Serratia marcescens is about 90% of the time a gray organism on blood agar, but occasionally Serratia marcescens can take up a mutation where the organism actually becomes brick red. And then finally, you have to understand bacterial genetics when learning about new diagnostic tests like polymerase chain reaction or PCR or other molecular techniques. The ultimate goal of bacterial cells is to make proteins that form cell structures and function and to transmit that information into the next generation. Bacteria thrive to make proteins from cell structures and transmit them into the next generation, so their goal ultimately is to make babies. Okay? This information is transferred from cell to cell in the form of encoded DNA, and RNA is the blueprint for that protein construction. The protein construction undergoes 3 steps before binary fission, including replication, transcription, and translation. Replication is when you have chromosomal DNA that is duplicated and put into a daughter cell. Transcription is when single-stranded RNA is made from one strand of DNA, so RNA here is the template. And then in translation, this is the making of specific RNA from the messenger RNA code. This is the making of a specific protein using messenger RNA as the template. So how does genetic material multiply or double to be used in binary fission and then given to the next generation of cells? It undergoes replication, transcription, translation, and termination. Replication is when DNA is duplicated and put inside the daughter cell. Transcription is when RNA is made and complementary to one part of DNA. Translation is making the proteins, and termination is the endpoint. Okay, so DNA replication is a 3-step event: initiation, elongation, and termination. In initiation, energy from ATP begins to unwind the double helix of DNA. After initiation becomes elongation, where each strand of an enzyme is separated, becomes a template. DNA polymerase III moves along each strand and inserts new nucleotides or purines and pyrimidines. And then during termination, the 2 DNA replication forks meet 180 degrees later. They are separated from one another with enzymes, and each daughter cell will inherit one complete chromosome. In RNA transcription, the DNA double helix is unwound and single-strand RNA is made from the DNA single-strand template. This is when DNA is unwound and various types of RNA are generated in information transfer. This is also known as helicase, which is the enzyme. RNA polymerase is a combination with a promoter unwind the helix and separates the 2 strands within the gene. And again, that happens with the enzyme helicase. The enzyme will move along the DNA template and add complementary RNA nucleotides. Again, this is a phosphate that's either a purine or a pyrimidine. Transcription leads to 3 types of RNA, including messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA determines which protein will be slotted into position during translation. Ribosomal RNA will serve as a framework for that structure, and transfer RNA transports specific amino acids to where it should be bound. Translation is the process of making the actual polypeptide or protein. At the ribosome, messenger RNA and transfer RNAs meet, bound with their appropriate amino acids. Transfer RNA aligns themselves with the opposite messenger RNA and brings the amino acids into position. A bond then forms between adjacent amino acids on the growing protein chain, and transfer RNA releases the amino acids and then returns it to the cytoplasm to bond with other molecules needing transfer. That brings us to termination, where stop codons, which are 3 nucleotides forming a genetic code, stops translation. The stop codons terminate the addition of amino acids to a growing polypeptide chain. Extra chromosomal elements are things that we need to talk about, and one such extra chromosomal element is something that originates outside of the nucleoid would be plasmids. Plasmids are extra genetic pieces. They are small and circular, and they are pieces of double-stranded DNA. These plasmids are not essential for bacterial growth. Okay, what the plasmid does is it promotes antibiotic resistance and miscellaneous virulence factors of bacteria. They are encoded within these plasmids. So MRSA and VRE form from plasmid information. Plasmids may be transferred from cell to cell through what's known as conjugation via the conjugation pili or the conjugation pilus. So mutations are a permanent change in an organism's DNA, and they occur in several ways. During translation, an incorrect amino acid may be inserted in the polypeptide because messenger RNA was miscoded, or mutations may be the result of a physical element like ultraviolet light or chemical elements. Mutations may be the result of something spontaneous or induced during translation, like base pair substitutions or base pair deletions. And mutations result in organism expressions variability. For example, like I already mentioned, Serratia marcescens should be a gray-colored organism, but because of a mutation, it could actually appear brick red. Now, there can be several good and bad consequences because of mutations. For example, the Black Death, which is caused by Yersinia pestis of the 14th century, killed 25 million Europeans, and that was because of genetic mutations. In 2002, Joseph Heinebusch and his team of researchers proved that the gene carried on a plasmid of the new Yersinia pestis allowed it to survive in the midgut of the intermediate host, which is the flea. This organism was genetically altered by acquiring the survival gene from an unrelated organism. The plague then was easily transmitted from person to person because the flea would be able to take a blood meal. The flea then regurgitates approximately 25,000 plague bacteria into the bloodstream of the victim with every bite. And that was all because of a mutation. Yersinia mutated and no longer died inside the gastrointestinal tract of the flea. Some mutations are beneficial to the host and helps with survival and adaptation, but other bacterial mutations can be problematic. People that have sickle cell trait are less likely to be affected by malaria. And that's because people that have sickle cell trait have mutated to be less affected by malaria. The organism Plasmodium is unable to survive inside the sickled cells. This mutation is common in sub-Saharan Africa, where malaria just happens to be endemic. So, of course, the population of people there have mutated and have actually developed sickle cell trait, and so their sickle cells are not susceptible to the Plasmodium organism. Now, prokaryotes achieve genetic alterations and diversity through gene transfer. And gene transfer can happen in a couple of ways. One is called vertical gene transfer, and the other is called horizontal gene transfer. Vertical gene transfer is when the parent cell divides by binary fission and there's exact copies of genetic material separated by cytokinesis. In horizontal gene transfer, intracellular transfer of genetic material from a donor cell to a recipient cell is what occurs. Usually not in a traditional reproductive fashion, free-floating genetic material from one source gets inserted into the template of another source. For example, a eukaryote could engulf a bacterial cell, and the cell is normally degraded, but if genetic material remains inside of the cell, then that changes the template of the eukaryote. Genetic recombination and horizontal gene transfer is an intracellular transfer of DNA from a donor cell to a recipient cell. Genes are transferred by various mechanisms to other cells of the same generation. Genes are then transferred between similar regions of two DNA molecules, and those DNA regions in the recipient cells are replaced with new genetic information. This is what happens when certain strains of bacteria become resistant to antibiotics. Genetic material may be horizontally transferred from one organism to another in three ways, and those three ways are called transformation, transduction, and conjugation. Transformation occurs when genetic material is introduced to another bacterial cell by horizontal gene transfer. The organism takes in foreign DNA and then starts to express that gene. And in this diagram, we can see an example of transformation where we have DNA fragments, and those DNA fragments get introduced into another bacterial cell by horizontal gene transfer. The organism takes in the foreign DNA and it starts to express that gene. When DNA is transferred from one bacteria to another by a virus, this is called transduction. This is the transfer of bacterial genes by a bacteriophage, which is a virus that carries the chromosomal DNA fragment from a donor cell to a recipient cell. Viruses like the macrophage have a protein coat and DNA or RNA within. The phage comes along and injects its core material into the cell. The original DNA is destroyed, but the phage genetic material replicates inside the cell. The cells are going to last, releasing new phages. Some bacterial cells have their genetic material integrated with the DNA in the phage. These cells undergo binary fission, and the genetic material is then transferred to future generations by vertical gene transfer. Some of the phage or bacterial DNA hybrids produce toxins like the ones in Corynebacterium diphtheriae. In this diagram, you can see a viral bacteriophage that attaches to a bacterial cell. The phage then inserts its DNA into the bacterial cell. Transcription, which is RNA made from DNA of the phage, DNA occurs, and then genetic material of virus and bacteria end up as sort of a genetic soup. The virus is then reassembled inside the bacterial cell, then released, and genetic information is now altered. When genetic material is transferred by direct cell-to-cell contact through the conjugation pilus, this is called conjugation. Genetic material is transferred from cell to cell in cells that are in close contact with one another. The close contact of the cells comes in the form of the conjugation pilus. This sex pilus binds to the recipient cell and then reels it in. Plasmid DNA containing the F factor for fertility is then transferred to the F cell via the hollow conjugation pilus. In this diagram, we can see the pilus of the donor cell attaches to the new cell. A piece of genetic material is transferred to the new cell via the conjugation pilus. Genetic material is joined and both cells now have changed and are viable donors of new genetic material. Restriction enzymes are used to restrict foreign DNA from hijacking the genome. In genetic engineering, a restrictive enzyme is kind of used like molecular scissors. We cut open the prokaryotic chromosome or a plasmid at a specific location so that the genetic engineer can insert a DNA segment from another organism. Enzymes are going to restrict the replication of a specific genome. We can manipulate the genome by inserting whatever we want into the lagged genome. Some examples of products derived from genetic engineering would be alpha interferon, okay? And this is because of the use of restrictive enzymes as well. Alpha interferon is used with antivirals to fight viral infections. Alpha interferons is made by WBCs, and WBC's genetic code can be modified to make more alpha interferon. So it's used as a viral or tumor treatment. Factor VIII is made because of genetic engineering and restriction enzymes as well, and it's used to replace clotting factors in people that are hemophiliacs. Factor VIII can take certain mammalian cells and genetically alter to make high volumes and high pure forms of factor VIII, and we can use that factor VIII to treat hemophiliacs. Tissue plasminogen activator is another product made from genetic engineering or the use of restriction enzymes, and TPA can be given to stroke patients to prevent further strokes or break down blood clots. And then another example of genetic engineering or the use of restriction enzymes would be epidermal growth factor, which promotes wound healing, especially in burn centers. So that's it. That's Unit 3, where we talked a little bit about microbial genetics. We talked a little bit about the pioneers of the genetic world. We talked about DNA and RNA, and we talked about translation, transcription, and termination, mutations, and also gene transfer. So those are all the things we covered in Unit 3. And here's just a little tip on how to be successful. Accept your personal responsibility and see yourself as the primary cause of your own outcomes and experiences. So in other words, you have the power to be successful. You just got to reel that power in.